نرجع كان هدف what is secreted to the intestine from the liver. كان هذا هدفنا intestinal secretion, biliary secretion to the liver, and this biliary secretion will come to the bile duct from hepatic cells. حكيت شوية عن anatomy اللي بيهمني. So these cells, they secrete their content, these canaliculi, collected into the bile duct, hepatic duct, right, left, etc. تفاصيل anatomy. بننتهي ب common bile duct. The liver cell, they have many functions, but their secretion will go metabolic function to the portal vein, to the systemic circulation. What is secreted here, what we call it bile. So what is the constituent of bile? Why we need bile? We have one essential element which is needed for fat digestion. So bile contains many components, and these components, what we have the bile salts, what concerns us. Cholesterol is a waste product. Lecithin is another variable which are essential for what we need. And we have other components, many other components. We have waste product. We have bilirubin. حكينا عن bilirubin لما شرحنا blood metabolism of blood, metabolism of red blood cells. What concerns us now, the volume, bile salts, lecithin, and cholesterol. Why we need that? We need that because we want to emulsify fats, to make fat soluble. Because all the reactions in the intestine, they are water medium. And fat is insoluble, water insoluble. And lipase cannot digest fat unless it is in water medium, it is soluble. How we make it soluble? By emulsifying that product, that fat, while mixing with the bile salts and lecithin. The next subsequent step for this is to help in make this molecule soluble to be transported through lymphatic system after absorption. بنحكي عنه بعد الامتحان عملية absorption and the importance for absorption. So we need it for emulsification before digestion. They are linked together. If we don't have emulsification, we have very minimal digestion. And we need to solubilize to form micelle for absorption process. That's why we need these biliary secretion. So these biliary secretion are essential element for bile salt secretion, essential element for also other waste products. نشتغل حاليا في other waste products. إحنا حاليا في digestive process, fat digestion and absorption. So this is the major function. So what we are talking about, this is the bile salt, which is a derivative of cholesterol. ضمن البايوكيمستري بيشرحوا تفاصيل السينثيسيس، هون ما بيهمناش السينثيسيس، بس we have to understand it is synthesized from cholesterol and it is a waste product. So it is essential for what we call it emulsification of lipid. And you know the detail of emulsification. You know, make it so in solution. This is the way this is the description. The essential component of fat with bile salts, lipids, and lecithin to make this molecule from water insoluble to water soluble molecule. And once it is water soluble molecule, lipase can work on it, and micelle to break down the triglyceride to release fatty acids and to help in absorption. So this is the way we are talking about. So the material will be secreted, going down through the hepatic duct to reach the intestine. كان امبارح كنت احكي لكم ننسى حاليا. بدي ارجع لها. What is this? This is a structure. It is extension from the bile duct, or we have the common bile duct, the hepatic duct. We call it the gallbladder where we have cystic duct. What's the value of this? It is a storage area, storehouse, only storehouse. So biliary secretion, usually all the time, 24 hours. We have certain rate of biliary secretion. This biliary secretion will increase during meal. And what happens during 24 hours? In between meal. In between meal, the material secreted will be stored here. And storage here, it has another function, to concentrate. It will reabsorb water and reabsorb other elements and concentrate the byproducts. The origin طلع what's the concentration of that material secreted and what happens after they are stored in the gallbladder. Just imagine we are talking about concentrating bile around 6 times. It is the same. What is coming from the bile, from the liver, will reach the gallbladder. What leaves the gallbladder? This is the concentration of what leaves the gallbladder. So we are talking about concentrating bile salts. And what we have, cholesterol, it is also concentrated, and lecithin. So we deliver to the intestine a material at high concentration after meal. Yeah, we store it and deliver to the small intestine immediately after meal when we need it. A small amount in terms of volume, but with very high concentration of bile salts to make life easy for lipase, to solubilize the fat, to make it easy for digestion. So this is the bile, which is coming from the liver and the gallbladder, the only storehouse. So can we live without gallbladder? So what happens if we remove gallbladder? Can we live without gallbladder? ممكن نجاوب عليه كمان شوي. So this is the function of the gallbladder. How we empty? How we pump this concept? قبل ما نكمل how we empty this content, how we pump this, we have secreted material. It will come to the small intestine here, and it will help in digestion. What is secreted from bile salts, at the end, it is reabsorbed and goes back through portal circulation back to the liver. So we have what we call it enterohepatic circulation. So bile salts, they are synthesized here, secreted, stored, or secreted directly here, will help in emulsification, no effect, no digestion process. Some of them will be lost through the intestine. In the terminal area, these bile salts, they are reabsorbed back through portal circulation back to the liver. Now the liver, this circuit of bile salts, we call it enterohepatic circulation. What's the function of it? To keep using the, زي ما حكينا عن recycling of iron لما حكينا عن red blood cell synthesis أو amino acids برضه في red blood cell synthesis and a recycling process. Now what happens if we lose the terminal medium? سبق وحكينا what happens if we lose the terminal medium in terms of effect on intrinsic factor and B12 absorption. The same story, bile salts will not be reabsorbed. So we lose bile salts in the stool, in the feces. So what happens if we lose that? The liver will start to synthesize more and more of these bile salts. So at rest, or as in normal condition, the recycling of bile salts will make minimal amount of bile salts newly synthesized, around less than 10%. A small amount loss. But if we have any disease in the terminal area, we have more activity for the liver to synthesize bile to replace what is lost. Now, how we empty gallbladder and how we pump the contents. نرجع للأساس. What comes from the stomach to the small intestine, it will stimulate, it is acidic, partially digested material. Acidic will stimulate secretin, حكينا عنه امبارح. Partially digested material, it will stimulate cholecystokinin. it will stimulate gastric inhibitory peptide. حاليا بيهمناش gastric inhibitory peptide, حاليا بيهمنا cholecystokinin. What is the function of this cholecystokinin? To stimulate one element to reduce gastric motility, but the more important element في عندنا to stimulate pancreatic enzyme كان البارح عنه كلمتين, و to stimulate the biliary tree. Three biliary tree gallbladder and the common bile duct contraction. The effect of that to empty the gallbladder. So immediately after meal, due to CCK, which is due to partially digested material coming from the stomach to the small intestine, it will secrete this cholecystokinin and goes to circulation to stimulate the biliary tree to secrete the content of the gallbladder to increase secretion from the liver, and this will increase the material coming in the bile, the content of the bile source coming to the liver, and we have the other effect, it will relax sphincter folding, lower sphincter at the junction of the common bile duct with the second part of the liver. So it will relax this and allow contraction, so the content will be delivered to the second part. So the second part of the small intestine will receive what comes from the stomach and the first part of the liver, what comes from the pancreas and what comes from the biliary tree to complete digestion, to complete the process of digestion. طيب, how we regulate that? نرجع نشوف how we regulate that. So we are talking about during peristalsis, we are talking about nervous control, نرجع nervous control. In addition to hormonal, so we have hormonal, we have nervous control, which will stimulate the secretion of the spine. Do we have other function of the liver? طبعا في عندنا عشرات الفانكشنز اللي ما حناش دخل فيهم الميتابوليك. But في عندي other function, secretion of cholesterol. في other function حكيت عنها من شوي صغيرة, secretion of bilirubin. في other functions, secretion of many waste products. Cholesterol is one of the elements which is secreted by bile, and it is needed for emulsification process. برضه مش ضمن اختصاصنا نحكي عنها ضمن البيوكيمستري. بس it is our concern to talk about that. It is part of the secreted material in the bile, where we are talking about the cholesterol. So we have, these are the secreted material. We concentrate on bile salt, we concentrate on lecithin and cholesterol for digestion process. Bilirubin and others for waste products, and etc. etc. هذا اللي بيهمنا من secretion تبعت البيليار secretion هذا enterohepatic circulation اللي وصفتها من شوي صغيرة. Most of the bile salts excreted in the duodenum are reabsorbed. Where they are reabsorbed in the terminal area, and زي ما وصفت من شوي صغيرة, any pathology in the terminal area or removal of the terminal area will interfere with this recirculation. This interference with this recirculation will put a load on the liver to produce more and more of the bile salts. If we lose it, but if we retain it, we preserve or we reduce the ability of that. So liver makes new bile salts to replace طبعا any more loss, the liver will produce more of these bile salts. هذا هو السكريشن. لحد الآن ايش اللي وصفنا؟ وصفنا gastric secretion، وصفنا pancreatic secretion، وصفنا biliary secretion. ضل عندي جزء أساسي اللي هو intestinal secretion. Is it important? بنتذكر لما حكينا to fulfill the requirements. اتفقنا لما حكينا عن البنكرياس، حطيتها كمان شوي، حكيت liver secretion، هي حكينا عن liver secretion. بعدين بدنا نيجي ل intestinal secretion. Intestinal secretion. What are these intestinal secretion? If we look at the structure of the intestine, what do we have? We have certain mucous membrane. هناخد بعد الامتحان لما احكي عن absorption بيهمني أكثر الستراكشر of the small intestine, membrane of the small intestine. في عندي villi, في عندي microvilli. In between these villi and microvilli, we have certain structures, what we call it, crypts of Lebercon. These crypts, where they secrete, they have mucus cells. In addition, what we have Brunner's gland in the small intestine. They secrete also mucus. They secrete mucus, and that secretion, كل secretion they are alkaline, ما عدا الأسيدي, والسليبري almost neutral. اتفقنا سابقا. So, what controls this secretion of the intestine? What controls this? نرجع للأساس. Distension, presence of partial digestive material, and the neural control. So what's the function of the mucus? Again, what controls that? The neural control, which increases secretion. So do we have other secretion from the small intestine? نرجع للهيستولوجي, المايكروهيستولوجي of the small intestine. اتفقنا في عندنا certain cells on the epithelial cells, في عندي villi, في microvilli. These, the membrane of the microvilli of these cells, they are desquamated during digestion due to exposure to the material. اتفقنا ضمن الموtility, لا بدي أرجع للموtility. حكينا الموtility of the small intestine, it has two concepts. One concept, the peristaltic movement. One concept, mixing. One concept, to expose the content to the surface. Why we want to expose the content to the surface? Because the surface will desquamate, one aspect. Desquamation of the surface will release the cells. ناحية تانية, exposure of this material to the surface will make a probability of absorption. ننسى probability of absorption تبع الامتحان. What happens if we have desquamated cells? These desquamated cells, what we call them enterocyte, they have certain enzymes. These enzymes, enterocytes covering villi, they have certain enzymes. What are these enzymes? نرجع للdigestion اللي حكى في البايوكيمستري بالتفصيل بشغل. هلا digestion in the pancreas, in the stomach, produce many polypeptides, maybe amino acids, but mainly polypeptides. What happens in the small intestine? The pancreatic function. The pancreatic function can produce all the way to monosaccharides, but it can produce disaccharides, many disaccharides. It can produce amino acids, but can produce also many dipeptides and tripeptides. The same story in fat, in terms of free fatty acid and glycerol. What they contain these? The membrane of the apical membrane of the enterocyte. They contain many peptidases. لازم نكتب dipeptidases or tripeptidases. So what happens if we have dipeptidases? It will break down the final, it has the final effect in the breaking down of the proteins. So to produce the final component, which is the single amino acids, we need the dipeptidases. And we have lipases, minimal amount of lipases. But we need another important element, disaccharidases. شو من disaccharidases؟ إلى saccharase, maltase, isomaltase, lactase. كل هذول they are essential part of the membrane of the enterocyte اللي هم they are covering these villi, microvilli. بس نحكي كلمتين عن اللاكتيز. برجع بحكي بالمناسبة هلا بعد الامتحان في 3 محاضرات فيزيولوجي. أول محاضرة راح أعطيها أنا، ومحاضرتين ثانية راح أعطيهم الدكتور محمد فايز، بدي أشرح تفاصيل الدايجستشن اللي البايوكيميكال أسبيكت. بس أتعدى عليه شوية صغيرة. اللاكتيز enzyme. This is an essential enzyme to digest lactose, which is found in the milk. And any deficiency in lactase enzyme will end in certain failure to digest lactose. اللي هو milk sugar. And anytime we have deficiency in final digestion, what is the end product? The end product, we have certain molecules in di components, disaccharides, dipeptides, although we have absorption of dipeptides, fat, or maybe. So what's the value of this? They are not absorbed easily. What happens if they are not absorbed easily? They work as osmotic factor. What's the meaning if they are working as osmotic factor? They prevent water absorption from the small intestine. So they keep certain volume in the small intestine. This is one aspect. And the second important bad aspect, زي ما حكيت كلمتين امبارح لما حكيت عن deficiency of pancreatic enzymes, they are good media for bacteria. Good media for bacteria. We start to have bacterial growth on these material. So what's the end? We deliver to the large intestine fluid which contains good nutrient material. Basically, we should deliver to the large intestine material almost free from amino acid, almost free from fat, almost free from carbohydrates. But if we don't have complete digestion, we don't have complete absorption. If we don't have complete absorption, we will deliver to the large intestine certain material of dipeptides, disaccharides, and certain fatty acid, which is a good media for bacteria to grow on it, and the complication of this bacteria, the distension, gases, and osmotic factor, where we will end in diarrhea. Another typical example, if we don't have these enzymes, the individual who have this lack of this enzyme, they cannot take milk as an example, because the milk sugar cannot be digested. نرجع نلخص نعيد الكلام. Intestinal secretion, we are talking about mainly mucus, alkaline mucus from the glands, سواء Brunner's glands أو سواء glands Krebs or Leberkan, certain glands there where we secrete the water structure, the fluid, huge amount of this fluid, and we have to concentrate on these enterocytes, the enzymes, which are the enzymes for the final digestion of carbohydrate, final digestion of peptide, final digestion of lipid. لا يعني إنه ما بصيرش عندي digestion, but it is not complete digestion. So we are talking about this. The failure of final digestion, we will end in delivering certain raw material, good nutrient material. We lose it, we don't benefit from it, but also we deliver it to the large intestine for one, it is a good medium for bacteria. أي سؤال لعندنا؟ أي سؤال عندنا؟ Okay. What about the large intestine? What about the large intestine? Large intestine في صورة سابقة نرجع لها. We deliver the contents أعتقد في الموتيليتي مش الموتيليتي. We deliver the content, they are fluid. Do we have nutrient material? الجواب لا. شوي زي اللي جاوبت عليها هذا السؤال. But what happens in the large intestine? We have water reabsorption. And we have some secretion of certain mucus to make mucus, the aim of the mucus to lubricate and to make things to move easily, to decrease resistance. So this we have certain cells to secrete mucus, but we have to remember we have no enzyme secretion. زي ما وصفنا في الشغل الثاني we don't have اللايم. زي ما حكيت بعد الامتحان هشرح عن الابسوربشن the role of the lime, microlyte in absorption to increase the surface area, which is essential for absorption. And these clips, they secrete alkaline mucus. And we have water reabsorption, some chloride reabsorption, mercury absorption, some bicarbonate absorption. And mucus secretion increased by parasympathetic. نفس الشيء حكينا لما حكيت عن small intestine. The parasympathetic will stimulate this. This is what we want to know about secretion from the large intestine. بهاي السلايد أعتقد إن خلصنا كل السكريشن concept from mouth to anus. From mouth to anus. What's the value of the secretion? What's the value of the secretion? What's the value of the secretion in different parts? The value at the end we need to digest. Why we need to digest? To make absorption easy, because absorption in the small intestine, most of the absorption is in the single structure format. شو single structure format? له single amino acid. But we will talk about dipeptide can be absorbed also. We want to talk about single monosaccharide, but we have some disaccharides can be absorbed. But mainly we talk about single monosaccharide which can be absorbed. Lipases, fat acid, it's the easiest one to be absorbed if we have the bile salts. If we have the bile salts, because they will form what we call it the micelle through the cell on the opposite side and the basolateral membrane side, and it will be drained through the lymphatic system. The amino acid والmonosaccharides they are drained through the portal vein اللي بنحكي عنه بعد. أي سؤال؟ أي سؤال؟ محاضرة سريعة. نفتح الباب النقاش. بكرة فيش محاضرة فيزيولوجي. دكتورة دينا بتحكي لكم امتى بتتعطي بالاناتومي اعتقد 9:30. بعد الامتحان المادة اللي داخل الامتحان بآخر كلمة اللي حكيت اليوم. أي سؤال؟ ما راح نترك بس أي سؤال؟ طيب نرجع خطوة هلا. احنا مشينا مشيت بالشرح مشيت لونجيتيودينال. أنا بدي أمشي هوريزونتالي أراجع كويكلي هوريزونتالي. نيجي الدماغ. What do we have in the mouth? بناء على معلومات الاستراكشر ننسى معلومات الاستراكشر. We have in the mouth skeletal muscles. All the muscles in the mouth they are skeletal muscles. They are voluntary muscles. But we have the concept of reflex. Reflex involuntary, سواء it involves skeletal as well as involuntary muscle, involuntary mechanism process. We put the material in the mouth. We break it down. We start initially by mechanism, we call it voluntary process. Then chewing process is reflexive process. Reflex is involuntary but uses muscles of mastication which are voluntary muscles. Next step we have swallowing, continuation in the motility process. This is also voluntary process, but it will stimulate cascade of reflexes. Cascade of reflexes اللي وصفناهم ابتداء من closure of the cessation of ventilation, closure of the nasopharynx, raising of the larynx, abduction of the vocal cords, and epiglottis will cover that. وبتبدأ muscles of swallow will be, the bolus will go to the pharynx and start the primary peristalsis. So in the mouth, do we have digestion? Which is the next step. Yes, we have digestion. Who breaking down? So do we have complete digestion? Chemical digestion? We may have chemical digestion. So why we don't have complete digestion? Because the time stay of the material in the mouth is very short. It's very short. لو نحط شوية carbohydrate and keep it in the mouth, yes, we will end in digestion. Amylase will break down. بس عادة material will stay for a very short period of time. So do we have absorption in the mouth? Yes, we have absorption in the mouth. Minimal absorption. We have certain drugs that are absorbed in the mucosa. So the mucosa of the mouth can absorb, but we don't have enough time for the material to stay in the mouth. The secretion in the mouth can digest, but the material cannot stay for a long period of time. So when you swallow, the lateral constrictor muscles contract, superior, middle, inferior, حتى to bring the bolus to the esophagus. Esophagus اتفقنا upper third skeletal, middle, lower, smooth muscle. اتفقنا concept آخر وين مكان. Any change in muscle length will reflexly make a reflex to oppose that change. Yeah, so I'm shortening to oppose it in elongation oppose it in shortening. Distension means we are talking about elongation of the muscle. So this elongation of the muscle, distension, will stimulate contraction. وما ننسى في عندي reflex أو آسف primary peristalsis جاي لي من constrictor muscle. So it will stimulate اللي هو we call it peristalsis. And we define peristalsis as contraction preceded by relaxation integrated by myotonic nervous plexus. في عندي proximally we have contraction. Proximally a reference عندي mouth. Distally away from the mouth. So we have contraction preceded by relaxation. So this will progressively bring the bolus down. What's the function of esophagus? One thing, conduit, to bring the material, يوصل من محل لمحل, from the esophagus, from the pharynx to the stomach. Do we have digestive function? No. Do we have secretory function? بس mucus. Digestive function اللي هي استمرارية للي بيحصل بالماء. بس time stay very short. حياتهم ما بنحكيش عن الدايجست. بس ما يقصد الاسوفيجاس في upper esophageal sphincter و lower esophageal sphincter. How they are post sphincter عرفنا area of high pressure. It allows movement in both directions according to the pressure regulator. طبعا during swallowing, one of the reflexes to relax the upper esophageal sphincter to reduce pressure. So the content will go from the pharynx to the upper esophageal, upper part of the esophagus. تمشي البريستالتيك ويف through vagus and local factors في inhibition للlower esophageal sphincter to allow opening of it to send the material to the stomach. أيضا في receptive relaxation in the upper part of the stomach اللي هو الفندس and the body. What are the functions of the stomach? Motility, yes, we have storage, we have mixing, we have emptying. With emptying, we have mixing also. Do we have secretory function of the stomach? MashaAllah, and the secretory function. The stomach will secrete many things to the lumen, to the mucus, to the intrinsic factor, to the physiology, but it secretes to the interstitial tissue, histamine, to the circulation gas, to the circulation gas. حكيت hydrogen ولا لا to the lumen؟ ما حكيتوش برجع. So we have this secretion. What's the function of this secretion? To break down mainly proteins. Do we have carbohydrate digestion? Yes. Why? Simply because the time, the receptive relaxation will last for maybe up to an hour, less than that, will keep the material coming from the mouth in status quo, no change. So the activity of the enzyme is there. But after a while, we start to have secretion of acid, which shows the pattern of secretion of acid. The pH will start to change, and the effect of the amylase will be suppressed, will be broken even, suppressed, and it will be digested. So what's the value of this? Acid will start breaking down protein. Protein pepsinogen will be secreted, mechanism due to parasympathetic stimulation, and will end more pepsinogen and acid, and digested product will stimulate more and more secretion of acid. This acid ends in relatively certain degree of breaking down some amino acids, many peptides, many larger peptides. كان عند regulation كان في عند cephalic phase، نفس الشيء cephalic phase للcerebral secretion، نفس الشيء cephalic phase للgastric secretion. الarm of this cephalic phase اللي هو vagus nerve. This vagus nerve will send information to the stomach to perform effect. اتفقنا vagus nerve will stimulate acid by itself, stimulate gastric, and stimulate enterochromaffin like cell, which stimulate histamine, and all of them they stimulate acid secretion, which will help in directing the breaking down the proteins. طبعا خلال الفترة هاي بتبدا الاكتيفيتي تاع السtomach تزيد. في عندنا البيزل الانتريكال ريتم اللي هو حكينا starts in the mid and try to proceed. As it proceeds, it will get more activity, stronger, and the time between subsequent waves will be short. So we get stronger contraction. The structure تبع السtomach, the fundus and the proximal part, we have muscle layer. But as we come to the virus, we have relatively much more muscle layer. إيش معناته much more muscle layer معناته force of contraction. As the contraction comes from the body or fundus down to the virus, the force of contraction will increase. It will try to push with it certain food particles. Some of it will leave through viral canal, but the viral canal almost it will close. And the rest will go back, and this will help in mixing. حكينا عن secretion, حكينا عن digestion, حكينا عن motility. Do we have absorption in the stomach? We have minimal absorption. We don't concentrate on that. Certain drugs can be absorbed. Amino acids, rarely. To have absorption, we need certain environment. We need membrane. We don't have that in the mucosa of the stomach. نفس الشيء we don't have that in the mucosa of the mouth. But we have minimal absorption. عشان هيك كان في عنا minimal absorption. نيجي ل small intestine. That's the key area where we have completion of digestion, completion of absorption of small intestine. But we have motility for mixing. We need motility to bump the content down. We need the motility to expose the content to the surface for absorption. The small intestine will receive the acidic material, will receive the pancreatic alkaline material with enzymes, will receive the biliary alkaline material with bile salts. Bile salts, enzymes, in alkaline media will complete digestion, will digest the material. The alkaline will neutralize the acid which is coming from the stomach. The alkaline which is coming from the pancreas and the alkaline material coming from the biliary. Now we have the small intestine will have huge area. Although the diameter is small, we have huge area in terms of villi and microvilli. This huge area will make good surface for absorption. This huge area, the microvilli, they are covered with a certain cells, we call them enterocytes. These enterocytes, they have in their membrane, structurally, they have enzymes also. So the mucosa of the small intestine is secretory of mucus. The mucosa is secretory for certain enzymes, for final digestion, اتفقنا diabetes and disaccharides and certain lipases, and they are good for absorption. And the small intestine is good for contraction, اتفقنا, for mixing, for pumping, and for exposure to the cell. In the small intestine, we have complete absorption, almost complete. Then we have to get rid of the leftover. شو اللي left over, how we will get rid of it. اتفقنا في عندي certain kind of motility, what we call it, ايش we call it, migrating motility complex. Try to evacuate. اتفقنا في عندي two mass movement. كل الموفمنتس اللي وصفناها it involve reflexes for short distance. إلا ثنتين اتفقنا عليها. واحدة migrating motility complex والثاني اللي بدي احكي كمان شو كلمتين عنها شو اسمه mass movement in large intestine. So emptying of that. But at the end of the small intestine في عندي sphincter في عندي valve. The valve will open toward the cecum. The sphincter is high pressure area. So we have to reduce high pressure area if the small intestine pressure will increase. And that's what happens. Due to rising pressure in the small intestine during migrating motility complex, reflex will inhibit the sphincter and the valve to allow it to open, and the content will move from the small intestine from high pressure area to the cecum of low pressure area. خلصنا هذا الشيء. بصير the cecum is high pressure area. High pressure intern, high volume. So it will try to contract. If it will contract, some of the content will try to come back through the ileocecal valve. But the ileocecal valve will close if we have high pressure in the cecum. If we have high pressure in the cecum, reflexively the pressure in the valve will increase above. So we have increased pressure in the cecum, but the pressure in the ileocecal valve will be more. Like the probability go back is not there. So the content will go through the ascending cord. Through that, we have certain mechanism to have contraction of long segment. This contraction of long segment will help in, أو قبل ما نحكي عن contraction of long segment, in the large intestine it will receive fluid to semi-fluid constituents. We have absorption of water, we have absorption of bicarbonate, chloride, and we end in solidification of the content, more solidification as we go through transverse colon and ascending colon. Now the contraction method, the structure of the large intestine, اتفقنا انه الاوتر لونجيتيودنال مش مغطي كل اللايرز. في عندي تينيا 3 cluster of 3 bundles وفي عندي circular muscle. Due to contraction at the same time يصير عندي appearance of the ostral, والostral appearance حكينا is not permanent appearance. It is permanent appearance but not permanent location. So the location will change. Now, the kind of motility in the large intestine involves long segment, which propel the contents for long distance until it will bring the contents to the sigmoid colon and to the rectum. Any distinction will stimulate, there is something similar to bristles. In the rectum and anal canal, once we have distension, it will stimulate myenteric reflex. اتفقنا في عندي myenteric reflex, contraction, distal inhibition. نرجع ل last part of the large intestine. اتفقنا كله في smooth muscles من second part of the esophagus to the anus في عندي smooth muscle. بس the first part of esophagus mouth skeletal muscle. Around the last part of the anal canal في عندي smooth skeletal muscle. Last part هو smooth muscle بشكل sphincter وسميناه internal anal sphincter. Anytime we have distension to stimulate contraction, myenteric reflex, and external anal sphincter will relax. ايش يعني will relax? Pressure will decrease. So the content will try to leave. This is what happens. This is the normal natural reflex. This is what happens in newly born individual. Anytime we have constant coming, opening, and defecation. But we have anatomically another structure, external anal sphincter, and it is voluntary and can be trained. And once it is contracted, it overcomes the internal anal sphincter. ايش يعني؟ it will squeeze it prevent it. مظبوط هذا فتح بس البراني في بابين، باب الاول وباب الثاني، الباب الثاني مسكر. البحريه الامريكيه والايرانيه، الايرانيين مسكروا والايرانيين مسكروا، الامريكان نفس المنطق. لازم الاثنين يكونوا فاتحين حتى تمشي السفينه. نحن هون بنحكي عن براز نفس الشيء، شيء كثير. اوكي let's continue. هلا باي تريننج ايش اللي بصير عندنا؟ According to the environment, the external anal sphincter will open if and only if the environment is good. حسب التريننج تبع هذا الشخص. So reflex, how many reflexes we have? Early in life we have 3, 4, 5 times per day. With age, we may have 1 to 2. With the training, it depends how you train yourself. Usually 1 or 2. So accordingly, the external anal sphincter will open or close depending on the internal anal sphincter. هلا الريفلكس اتفقنا دائما شغال anytime we have reflex. بس الاوبننج تبع external anal sphincter is regulated by higher center. هذا يعكس ايش؟ نرجع للاوريجن انفورميشن اللي اتفقنا عليها. في سنسوري نيورونز بتوصل للماينتريك نيرفس بلكسس، في سنسوري نيورونز بتوصل للسباينال كورد، ومن السباينال كورد all the way to higher cranial. هذا الملخص كل شيء في الاناليسيس. ضل عندي الابسوربشن، حكينا بنحكي عنه اول محاضره بس مش اكثر من هذا، اللي هي اول محاضره بعد الامتحان الاربعاء القادم ان شاء الله. اي سؤال؟